Welcome to Lecture 2. We'll be looking at sections 1.3, 1.4, 1.5, 1.7, and 1.8. This will be covering the different cell types as well as viruses. The outline for today's talk is we will start with the differences between prokaryotes and eukaryotes. Then we'll move into types of prokaryotic cells as well as types of eukaryotic cells. Then we'll start to move into the bigger picture, model organisms. Again, we'll scale it back down. And since we've talked about prokaryotes and eukaryotes, we're going to look at cell size and how these cells differ. The next topic is synthetic biology, and then we'll end with viruses or viriyoids. The very last part, as always, is going to be an overview and then some keywords and terms for you guys to know. So with the invention of an electron microscope, scientists were able to actually determine and examine the internal structure of a variety of cells. So we'll see a little bit later when we look at cell sizes why we need an electron microscope, because some of these structures are extremely small for us to see. By using this electron microscope, we're able to create two fundamentally different classes of cells. We have prokaryotic cells and eukaryotic cells. They are distinguished by not only their size, but their type of organelles. Now, an organelle means little organ. So if you think of a cell as being like a little person, the organelles, little organs, are going to be exactly that. They're going to be the little organs of the cell. They're going to make the cell do all of its functions and jobs. So there are even little organelles called lysosomes that act as sort of a stomach and digest things. For prokaryotes, these are all bacteria. And eukaryotes include protists, animals, plants, and fungi. Eukaryote means true nucleus, so eu means true, and karyote means nucleus. So our bacteria do not have a true nucleus, whereas plants and animals, fungi, protists, amoebas, things like that, all have a true nucleus. Now we're going to start comparing our prokaryotic and eukaryotic cells. There are features that are held in common by these two cell types. Both of these cell types have a plasma membrane, and they're a very similar construction. The genetic information that's held within the DNA is coded using this identical genetic code, that A, T, G, and C. They have very similar mechanisms for transcription, translation of our genetic material. This includes their ribosomes. We have many shared metabolic pathways, things like glycolysis and the citric acid cycle. They have similar apparatus for the conservation of chemical energy. This is often in the form of ATP. They have a similar mechanism for photosynthesis. Now, our green plants are the ones that typically do photosynthesis in a eukaryotic cell type. But there is one kind of bacteria, the prokaryotic bacteria, cyanobacteria, that is also able to do photosynthesis, and we'll look at this a little bit later. Both of these cell types also have a similar mechanism for synthesizing and inserting their membrane proteins. And they also have proteosomes. These are protein digesting structures. This helps get rid of proteins that have gotten old or are misfolded in some sort of way. And these proteasomes are of a similar construction between archaebacteria and eukaryotes. But while there are some basic similarities between these two classification of cell types, because they're different classifications, they clearly have differences as well. This is a list of some of the main characteristics that distinguish prokaryotic cells and eukaryotic cells. First off is their complexity. Prokaryotes tend to be relatively simple, while eukaryotes are very complex in structure as well as function. For their cytoplasm, eukaryotes have they tend to have membrane-bound organelles, those little organs again, as well as complex cytoskeletal proteins. Cytoskeletal proteins are proteins that live in the cytoplasm, and they have sort of a skeleton-like function. So they're going to give the cell shape and movement. And so we'll look more in depth at these cytoskeletal proteins later, but just knowing that we have sort of a skeleton within these cells as well. One of the similarities here, though, is that both eukaryotes and prokaryotes have ribosomes, but they do differ in their size. For cellular reproduction, eukaryotes will divide by mitosis, while prokaryotes just divide by simple fission, meaning a bacteria will split completely in half and make two complete copies of itself, or two daughter cells. With locomotion, the eukaryotes have both cytoplasmic movement, meaning they can shuttle things within the cell itself from one spot to another. And the cell itself is also able to move at times, so we have cilia and flagella that will power this movement of these eukaryotes. Prokaryotes, though, have a flagella. Not all of them, but some may have a flagella, but they can differ in both form and the mechanism of how they work. And the last property that distinguishes prokaryotic cells and eukaryotic cells is their genetic material. Well, they have that same basic A, T, G, and C that encodes their DNA. The packaging is different. Prokaryotes just have this nucleoid region, so the DNA, that circular DNA that a prokaryote has, is just found compacted together in a nucleoid region, but it isn't membrane bound the way a eukaryote has. So we have that true nucleus that is membrane bound. And this is where all of our chromosomes reside. The amount of genetic material differs between prokaryotes and eukaryotes. Because eukaryotes are much more complex, they have different proteins, they're going to divide by mitosis, different locomotion, all of these things making them much more complex means they're going to need more genetic material. So there's much more genetic material than what we see in these prokaryotes. And lastly, the form of this genetic material is different. Eukaryotes have chromosomes made of both DNA and protein, while a prokaryote has a single circular DNA. This table, 1.1, compares, again, prokaryotic cells and eukaryotic cells. And it shows some features of these eukaryotic cells that we do not see at all in prokaryotes. So with the eukaryotic cells, they are divided into a nucleus and a cytoplasm. And this nucleus has it's membrane bound, so it has a nuclear envelope. And this is not found in a prokaryote. We have complex chromosomes composed of DNA and proteins. And this is used for mitosis to get compact together, as well as separate during this meiosis. We have these complex membranous cytoplasmic organelles, so these little organs, which include things like endoplasmic reticulum, the Golgi complex, lysosomes, endosomes, and things like that. Our eukaryotic cells also have specialized cytoplasmic organelles for aerobic respiration. This includes things like mitochondria, and then we have photosynthesis, where we use chloroplasts. Again, back to that cytoskeletal system, that skeleton for the cell. It includes things like microfilaments, intermediate filaments, microtubules, as well as proteins that will shuttle things back and forth within the cell. This is not found in a prokaryotic cell. Eukaryotes also have much more complex flagella and cilia than what can be found in a prokaryote. Plants tend to have cellulose in their cell walls. Eukaryotic cells also have 2 copies of their genes per cell, one from each parent. And they undergo sexual reproduction, so not fission, the way prokaryotic cells. So some of these things that we've listed here, I also talked about in the last slide as well, so some of these are reiterating as well as having a couple other things in there. So here I'm going to post a link in the materials section for Unit 1. And this is a video for YouTube in which we see sort of a cell doing its everyday thing. This is a eukaryotic cell. It's going to have microtubules. You'll see motor proteins carrying cargo within the cell. You'll see all kinds of things happening within it. And this is just meant to sort of give you an idea of the types of things that we're going to go through and talk about this whole semester. It's just a fun video to watch. You can revisit it later. It might help you put things in perspective as we start building on the individual cell structures. So I'm not going to play it here for you now, but I will post the link so that you can access that video and get an idea of what the cell does at any given time. Now we move into the structure of cells. So we'll start with the prokaryotes, which are bacteria. And these images of the structure of cells are just example images. It will have everything in it. So any structure that could possibly happen within a bacterium or a eukaryotic cell is going to be on there. It does not mean every bacteria has all these features or every eukaryotic cell has these features. It's kind of like taking the most fanciest car that there is with all the options and having it completely loaded, but we really know that in real life cars come in a variety of shapes and sizes with multiple different options, and most cars don't come with every option. So here with this bacteria, we can see the nucleoid region, so there is no membrane-bound nucleus. It's just a nucleoid region where all the DNA is compacted together. This is found within the cytoplasm. These little dots are going to be ribosomes. Bacteria can also have the cell wall. There's a plasma membrane, so we talked about the plasma membrane being similar between eukaryotes and prokaryotes. Some bacteria also have what is called a capsule around the outside. Some bacteria have a flagella. So these flagella are what are going to propel or aid in movement for these bacteria. And then there are also pilus or pili. They kind of look like hair-like structures on the surface. These pili are going to be used for attachment of a bacteria to some host cell, or attachment of a bacteria to another bacteria. The second cell type that we're going to look at for structure is going to be the plant cell. So this is a eukaryotic cell, but it still has very different structures compared to what we would say an animal cell has. So we'll look at an animal and a plant cell differently. Since a plant cell is going to be a eukaryote, it has a true nucleus. So here we have this nucleus. It has a nuclear envelope that is membrane bound, in other words. Then we have the rough endoplasmic reticulum. This is an organelle around the outside of the nucleus. Other organelles include things like chloroplasts, the smooth endoplasmic reticulum, mitochondria, a Golgi. And then we also have proteins like ribosomes. We have channels that go between these plant cells. Within our plant cell, we also have a vacuole. And then we have these complex cytoskeletal structures, such as microtubules. Now we'll look at an animal cell. Now we're going to have some of the similar features as what we see with the plant cell, like microtubules, but we also have other cytoskeletal structures, like microfilaments and intermediate filaments. These are often used within the flagella or in these microvilli to give the cell structure and also movement. So just like a bacterial cell, we can have flagella. We can have cilia as well. Cilia also can help with movement for these eukaryotic cells. Again, eukaryotic cell has a nucleus. Inside that nucleus, we have chromatin. This is where we have that condensed DNA or genetic material. Around our nucleus, of course, is that membrane, so nuclear envelope. Similar organelles again, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi, mitochondria. We also have ribosomes, proteasomes, centrosomes. We're not going to need to know all of these structures, but you do need to know the differences between animal cells, plant cells, and bacterial cells. So know a few differences between these and how to tell them apart. So this is sort of the basic structure of a cell. And this is a eukaryotic cell, and it's an epithelial cell, which is found in the male rat reproductive tract. So looking through this electron microscope, which I mentioned, this is how we were able to distinguish between prokaryotes and eukaryotes. With this electron microscope, we can see these small organelles. So we have rough endoplasmic reticulum here that kind of looks like ribbons within this cytoplasm. This is a dense area with chromatin in it. The nucleolus, which is inside the nucleus here. There are mitochondria, which are often sort of these kidney bean shaped organelles. We have a smooth endoplasmic reticulum here. The Golgi complex is here. Along the side, we have a plasma membrane to separate the cell types here. And then within the cytoplasm, there are cytoskeletal filaments, ribosomes, and things like that. So if you look at that YouTube video, you're going to see things relatively spaced out, and everything seems to have so much room to work and move and do their business. Well, it's not really completely true. Our images, our diagrams in these videos are going to portray the cytoplasm as being kind of this big open space. In reality, it is a very crowded compartment. So we're going to see here in red all of those cytoskeletons. So these are, like I said, the skeleton of the cell, sort of the bones of it. There's cytoskeletal networks everywhere. Ribosomes are everywhere. And here in blue, we're going to have cell membrane. So this high complexity of a eukaryote also comes with a lot of extra components that are going to actually fill up the cell. One of the differences between prokaryotes and eukaryotes that we talked about was their cellular reproduction. So we just looked at some of the structural features that are different between prokaryotes and eukaryotes. And now when we look at cellular reproduction, we see that our eukaryotic cells here are going to actually divide by mitosis. We have this mitotic spindle here in green, so these are microtubules that are going to pull apart the chromosomes. So in blue here is DNA in the form of chromosomes, and we split those two copies of our genes apart and make two daughter cells. Bacteria, on the other hand, typically undergo fission, so this bacteria would be able to divide completely in half, making two daughter cells, complete genetic copies of itself, and that would be the way it would typically divide. But bacteria also have the ability to share some of their DNA with another bacteria. So here we have a donor bacteria. Through a pilus structure, it's able to send some of its DNA to a recipient bacteria. In this way, some bacteria might be able to pass on other genetic material, things that might be advantageous to that recipient bacteria, like antibiotic resistance. One of the differences that we also talked about was that prokaryotic cells and eukaryotic cells both have a flagella, but structurally they are different, as well as the mechanism in which they work. So here we have a Salmonella bacterium. On this bacteria, it has multiple flagella. So not just one, but there are many flagella there. Multiple tails used for movement. If we look much closer, so that this is the inset, looking much closer, an electron micrograph image, we can see the structure of this flagella up close. For a human sperm cell, this is one of our cells that actually has a flagella as well, so giving these cells movement. If we were to cut that flagella, we would see this sort of structure in there. So these are different microtubules and things like that, cytoskeletal structure, to give it shape as well as allow for movement. Now, this is a human sperm cell, but we can see this same layout and structure in the flagella found in a protist or in green algae as well. So our eukaryotic flagella all look very similar, which are different from the prokaryotic flagella. So there are different types of prokaryotic cells. In fact, there are just two domains. We have Archaebacteria and the domain Bacteria. Within the domain Archaea, we have things like methanogens. They produce that methane gas. So this can be found in the bacteria that are found within a cow's gut or a moose's gut, which allows them to make methane gas. We have halophiles. So these are bacteria that love high salt concentrations. Then we have acidophiles. They are acid-loving bacteria. And then we have thermophiles. So these bacteria survive in high temperatures. Some of them can live at like 121 degrees Celsius. And knowing that 100 degrees Celsius is boiling, so they can live in boiling water. Within the domain Bacteria, this contains pretty much everything else. Starting with the smallest known cell, which is a mycoplasma, all the way to weird things such as Cyanobacteria. This is that photosynthetic bacteria. So they have these multiple membrane layers within them. And this is where the Cyanobacteria actually does photosynthesis. So it's thought that Cyanobacteria could have possibly given rise to our green plants when the atmosphere changed to a more oxygen-rich environment. There are also some bacteria that are capable of fixing nitrogen as well. This means that it will take nitrogen gas and convert it to something like ammonia, which can then be converted into things like amino acids, which are used to build proteins for the cell to use. Within this domain Bacteria, although I've named sort of different things, don't forget about the other bacterias that there could be. So there are things like disease-causing bacteria as well. Some are pathogens, some are non-pathogenic. Things that live within on our skin or within our gut all the time, which are non-pathogenic, and this is within the domain Bacteria as well. So with that, these bacteria have a huge variety of diversity. Prokaryotes are typically identified and classified on the basis of their specific DNA sequence. So if we get sick, we'll go to the doctor, they'll take a swab of your throat, they send it off to culture, and then they'll look and see what kind of bacteria grows. But whatever bacteria grows, maybe one of them has made you sick, maybe not. But not all bacteria live off of the same nutrients. So just like people eat different food, bacteria eat different foods. They live in different environments. Some like acids, some don't. Some like heat, some don't. They all have different requirements for growth and living. So by doing this culture, we may not have actually even caught the bacteria that is making the person sick. So the only real way to identify a bacteria is using DNA sequencing. And recent evidence has shown that prokaryotes are much more diverse and numerous than we ever really thought. So, we're looking at huge, huge numbers. 640 times 10 to the 28th number of prokaryotic cells overall. Many of these are found in the soil or within the ocean itself, or deeper within the soil, I should say. So there are bacteria not only living on us, but all over the earth as well. And like I said, some are pathogenic, some are not, most are not. But it makes up a good portion of our Earth's mass as well. So now we'll move into the types of eukaryotic cells. So a eukaryotic cell includes things like protists, animals, plants, and things like that. So we're often thinking about very complex creatures or organisms. But there are also unicellular eukaryotes, meaning it's a single-celled organism. While it seems like this would be really simple, very basic type of cell, think about everything that that single-celled organism has to do. Here we have Vorticella. It's a protist, and it's very highly complex. They have the ability to sense their environment, trap food, and evade predators, all in this one single cell. This is something that we have multiple cell types to do, and this is just one single cell, and it's able to do all of these different things. For multicellular eukaryotes, we have different cell types for different functions. So we have this cell specialization that happens, and it's called differentiation. So we all start off as these haploid cells. We have a sperm cell and an egg cell that get fertilized together. Together, they're going to form that diploid cell and undergo cell division. As these cell divisions keep going during embryonic development, the cells will differentiate into different types of cells. So we all start off as basically a cell, a single cell. The cell divides, and then they differentiate so that we get a wide variety of cell types to make up this embryo. This includes nerve cells, blood cells, muscle cells, fat, epithelial cells within our intestine or on our skin, bone, muscle, and also other connective tissue. And the way in which these cells differentiate or decide what to become is usually determined based on the signals that it would receive from its neighboring cells or the surrounding environment. So these signals are going to totally depend on where that cell is located within the embryo. So it's not as if right in the middle of these nerve cells we're going to make heart cells or intestinal cells, because they're getting environmental cues from other nerve cells that what they should become is a nerve cell or make up part of the brain. The number and arrangement of organelles relates to the function of the cell. So mitochondria, for example, are found in pretty much every cell type. In one cell type, though, they may have a much more rounded shape, but in another cell type, they may be highly elongated or even thread-like. It just depends on what the function of that cell is. And despite this differentiation, cells still have very common basic features, which you can go back and look at those basic features of an animal cell. So while we talked about looking at cell biology as typically sort of a reductionist view, we're looking at single cells and trying to apply this to a whole organism or a group of organisms or groups of cells. And that's very reductionist. While many of these cells have similar basic structures, we also then use an organism to model other organisms. So for a bacteria, in order to study bacteria, we have what we call E. coli. This E. coli we use because it's easy to grow. We have its genetic sequence already mapped out, and so we can easily manipulate it as well. We have many tools to utilize this E. coli and work with it. So this is what we would use for our prokaryotic model organism. You want to study some sort of cell biology aspect. A lot of times you would just go to E. coli, work with it, and then try to apply what you learned to other bacteria. Much of what we know, not only about prokaryotes, but also eukaryotes, in terms of replication, transcription, and translation, was founded by using E. coli as a model organism. The next one is Saccharomyces. This is a yeast cell. This is known as baker's yeast or brewer's yeast. It is one of the most basic eukaryotic cells that we have, but it has very similar proteins to what we have in humans. So some of these proteins have a very conserved function. Even though it may not be completely the same, we can look at what we see within the yeast and then try to apply it to some of our human cells as well. Arabidopsis, a mustard plant, it has a relatively small genome for a plant. It has a rapid generation time, it makes a lot of seeds, so it's easy to grow. They only grow to a few inches tall, so it isn't like we need a huge greenhouse for these to grow, making it very easy for us to culture and study. C. elegans is a nematode. This nematode has a very set, defined number of cells in it. It has about 1,000 cells, and they develop in a precise pattern during cell division. And so we can use this nematode because it's easily cultured. It has a transparent body wall, so we can study using fluorescent proteins. And we can use it for genetic analysis. Here they're looking at the nervous system of this nematode as well. So we can use things that don't seem very related, but it helps us understand the development of an organism as well. The next one, Drosophila, or the fruit fly, is a small but complex eukaryotic organism. We often use this in molecular biology because of its neurological similarities. So some of the proteins that we have found within Drosophila, just like within the yeast, Saccharomyces, are very similar to what we see in humans as well. We can genetically manipulate this Drosophila as well. And we can use it to study evolution and gene expression as well. The mouse is also small. It breeds very easily within the laboratory. There are a lot of different genetic strains. We're also able to genetically mutate them so that they can carry diseases like cancer, things like that, or diabetes. We've also modeled autism within the mouse. And so this helps us study human diseases as well. Now, again, we're going to take a step back. We've talked about these prokaryotes and eukaryotes and how they're different. I have brought up a little bit about electron microscopes. We've seen some images. Those are those gray images where you can see organelles and things. And so when we look at these prokaryotes and eukaryotes, it's hard for us to really imagine what their size is unless you've done some sort of laboratory research. So we normally measure a cell in a micrometer and nanometer. Micrometers are going to be 10 to the minus 6 while a nanometer is 10 to the minus 9. If we start with a hydrogen atom, and then you put, of course, 2 hydrogens and an oxygen together, you get a water molecule. So this is the size of an atom in a water molecule. Hydrogen atom being 1 angstrom in diameter. With an electron microscope, we're able to see things like DNA, the lipid bilayer of a cell, some of those cytoskeletal structures like actin, and we are also able to see things like viruses. The next type of microscope, a standard light microscope, so something you would be able to use in the lab. You can see things like bacteria, mitochondria, chloroplasts, simple cells like lymphocytes and epithelial cells, so these eukaryotic cells we can also see. And with our human eye, we can see things like a paramecium and a frog's egg. So single-celled organisms, we're going to often need a light microscope, while multicellular organisms we can see with the naked eye. The size of a cell is often limited, though. It's limited by the volume of the cytoplasm, and that is because that's what's supported by the genes in the nucleus. It is also, the volume is limited because it has to be supported by the exchange of nutrients. So we have also the distance over which these substances within the cell have to be able to travel. And sometimes within like a bacteria, it has to travel via diffusion. Epithelial cells, we have those cytoskeletal structures that will transport things, but we also don't want to transport them over a huge distance. So cells are going to be relatively small and compact and pack things in a highly organized, complex way in order to minimize the distances which these nutrients and other proteins need to travel within the cell. It wouldn't make much sense to have this giant, huge bacterium, but it's not able to actually collect and keep its energy or nutrients in the correct spot. And the next topic is synthetic biology. So, synthetic biology is a field that's oriented to create a living cell in the laboratory. Realistically, the idea is to develop a novel life form beginning with an existing organism. So, researchers around the world have attempted to genetically engineer organisms that possess metabolic pathways capable of producing things like medicine or pharmaceuticals, hydrocarbon-based fuel molecules, or other chemicals that, in a cheap way, made from cheap, simple precursors. Now, Greg Ventner and colleagues in 2007 kind of started this process, and they found that they could replace the genome of one bacteria with that of a closely related bacterium. So effectively, they changed one bacteria into another species of bacteria. So this is a way in which we're kind of like swapping the DNA and making one bacteria turn into another. And the last topic that we're going to cover today is, we'll cover viruses. Now, viruses are pathogens. They're responsible for dozens of human diseases. This includes things like AIDS, polio, influenza, our cold sores, measles virus, mumps, and even many different types of cancer. These viruses will occur in a wide variety of different shapes and sizes, as well as constructions, but all of them share this common property. They are intracellular obligate parasites. Now this means they have to live within a cell. Now keep note, this is intracellular, meaning it's internal, while the prefix inter, I-N-T-E-R, will mean between cells, so from one cell to another. But this means intracellular, meaning it needs to be within a cell to be alive, in a sense. A virus cannot reproduce without a host cell. Outside of the host cell, it's considered a virion. This is the virus particle outside of a host. Within the host cell, it's considered a virus. Now, the basic structure of a virus includes that it has genetic material, often a protein capsid around that genetic material. And some viruses may have a lipid envelope around the outside. This genetic material that a virus has can be single-stranded or double-stranded DNA or RNA. So while humans have double-stranded DNA and prokaryotes will have DNA as well, but in a circular form, viruses can be in the form of DNA or RNA. They can be single-stranded or double-stranded, so there's a wide variety of their genetic material and how it is encoded. Now, virions are different from viruses to some degree, and this is because they are a pathogen, but it is a small naked RNA molecule, so it's just RNA with no capsid protein coat around it. And they will cause disease by interfering with gene expression within a host cell. So while viruses have that same basic structure of this genetic material with some sort of capsid around it, viruses will look very different. Now, these three viruses that we see here, the adenovirus, bacteriophage, and HIV, their sizes are not equal here. We're just doing this for graphic purposes, but they are different in sizes as well. So the bacteriophage is a virus that will infect a bacteria. Of course, HIV is the virus that causes AIDS. And you can see each of these three have a different construction and shape. But on them, they have what we call surface proteins. So these are surface proteins, like here, GP120. These are surface proteins as well, and they will attach to the host cell. So these surface proteins are recognizing something on the host itself. And the way that we have specificity for a host, like this HIV or virus, doesn't infect a dog or a cat. And that's because this protein recognizes something within the human cell. So it is specific towards human. Now, there are other viruses, like rabies virus, for example, that can infect a variety of hosts. Things like dogs, bats, raccoons, humans. And that's because the protein that they're recognizing on those cells that they're infecting, the host cell, is common to all of us. So once a virus gets into a cell, we know that it has to be within the cell to, in a sense, be alive. So it's going to hijack the cell that it's in and create copies of itself. So, in a sense, the host cell that it's living in becomes a little virus factory. We use the machinery that the host cell provides in order to make a bunch of different virions. And in doing so, we see all of these viruses being produced within the cell. And then When there are enough viruses in there, sometimes we have a lytic infection in which the host cell then lyses or breaks apart, releasing more virus into the environment. Now, those viruses will go on and infect other host cells as well. Another possibility is integration. So some viruses, once they get inside the host cell, will put their DNA into the host cell's DNA or chromosomes. Now that host cell, or even the individual, the organism, may behave normally, and that is until some sort of stimulus activates that DNA, that virus DNA or provirus DNA, and then it leads to the lysis of the cell. An example of this are your typical cold sores or herpes viruses. So your herpes virus infects a cell. That DNA integrates into the DNA of a cell, of our cells. When you get very stressed out, or you get sick with some other cold or something else, your immune system is busy working, or it slows down, and it's not able to keep that herpes virus in check. The herpes virus then reactivates, and you end up with that nice cold sore on your lip, and that is the cell, or the virus is lysing the cell and releasing more virus. That's why when you have an actual cold sore, you're in a sense contagious. You're releasing more virus that you can give to other people and infect other cells. Another option, instead of lysing the cell, is that the host may actually just bud. So in a sense, the virus sort of pushes out through that plasma membrane, it coats itself in some of that plasma membrane from the host cell, and then moves on and infects another host cell. One of the bad things with integration is that sometimes these viruses will insert themselves into places in our DNA that causes the cell to become malignant. This is an example of how viruses can cause cancer. So HPV, human papillomavirus, is known to cause at least 16 types of cancer. And so that's why we have vaccines such as Gardasil in order to prevent that cancer, prevent the virus from infecting people, which then prevents the cancer. So if the virus infects the cell in a way, it can change the way our cells act. Meaning, maybe certain genes get turned on that normally aren't, making them divide and have uncontrolled cell growth, turning into a cancer or becoming malignant. So in today's lecture, we have similarities and differences between prokaryotic and eukaryotic cells. So look at that table 1.1 again. Go through some of these similarities and differences. We had complexity, cytoplasm differences, cellular reproduction, their locomotion, and genetic material. Organelles and other structures. This is found only in eukaryotic cells. So we have nucleus, mitochondria, ER, Golgi, chloroplast, things like that, cytoskeletons, cell walls. Those are found in eukaryotes. There are types of prokaryotic cells, like archaea and bacteria. There are different types of eukaryotic cells, and they undergo differentiation. So in eukaryotes, different cells will have different functions. So in a multicellular organism, a heart cell has a different function from a nerve cell, from a muscle cell, or a fat cell. They all have their own function. And then we looked at some model organisms. There were the 6 model organisms that we could use in different ways to study cell biology or molecular biology. And lastly, we covered some viruses, their structure, and viridoids as well. So that was just a naked RNA that acted in a way like a virus as well. And then the link we see here at the bottom is that link for looking at the cell, the life of a cell. So I'll post this link as well as a separate link outside of the lecture onto Blackboard as well, so you can see that. And the last thing that we have are some keywords and terms. We have organelles, or tiny organs. These are found within eukaryotic cells. They're prokaryotic cells. We have a nucleus versus the nucleoid region found in a prokaryote. The cytosol, or cytoplasm. Differentiation. This is how cells become specialized. We have viruses and virions, or virioids as well, which were naked RNA without any capsids. And then a provirus, and this is when the DNA of a virus integrates into the DNA of the host cell.